Lidström. I work as a postdoctor at Umeå University, and today I'm going to tell you about a study that we performed using the UCMP technology from Lumito. So my presentation is titled Galectin-4 mediates immune evasion in pancreatic ductal adenocarcinoma. So if you're interested about the results in this study that I'm going to present, you can find it in the January issue of Cancer Immunology Research. The study itself is titled Extracellular galectin-4 drives immune evasion and promotes T-cell apoptosis in pancreatic cancer. So pancreatic ductal adenocarcinoma makes up 85 to 95% of all diagnosed pancreatic cancer. It's characterized by late discovery. About 20% of patients are candidates for surgery, and another 5 to 10% are deemed borderline resectable. The median survival of pancreatic ductal adenocarcinoma is 5 to 6 months, and the 5-year survival is about 10%. So this is increased to around 20% with surgery, but because of the nature of this form of cancer, it metastasizes early, so it tends to recur with time. So a PDAC tumor consists of these tumor glands that are surrounded by cancer-associated fibroblasts. So these cancer-associated fibroblasts secrete extracellular matrix, which together with secreted factors from the cancer-associated fibroblasts and the cancer epithelial cells make up the matrixome of the tumor. The secreted proteins from the cancer cells has been found to affect metastasis and immunosuppression in this tumor. So the galectin family of proteins consists of 3 structural arrangements. You have the monomeric galectins, which makes up the majority of the galectin family. You have the tandem repeat galectins, where galectin-4 is a family member. And you have the chimeric galectin-3, which tends to form oligomers of very similar protein fragments. So the common function of this protein family is immunomodulation. So different galectins are expressed in different tissues, but all of them modulate the immune system. So specifically, galectin-4 is expressed in the GI canal, where it's involved in trafficking of glycoproteins in enterocytes. And it's also hypothesized to promote tolerance to food antigen because of its effect on immune tolerance. So galectin-4 has 2 distinct functions, which makes it a challenge to study. So you have an intracellular function and an extracellular function that's mediated by secreted galectin-4. When it comes to the intracellular function, it can increase differentiation in tumors. And this is based on studies of colorectal and PDAC tumors. So if you have a high expression of intracellular galectin-4, you tend to have a higher differentiation degree of the tumor. The extracellular function of galectin-4 is to induce T-cell apoptosis, and this is based on studies in IBD, on PBMCs in IBD, where they found that extracellular galectin-4 can bind to the CD3 receptor of T cells and induce apoptosis in that cell. So in this project, we purified the matrixome of PDAC tumors and compared that to normal healthy pancreatic tissue. And we found that a lot of proteins were overrepresented in the tumors compared to the normal tissue. Specifically, we found that galectin-4 was one of those proteins that were overrepresented in pancreatic ductal adenocarcinoma tumors. So to summarize the findings of this paper, we find that galectin-4 is produced by pancreatic cancer cells and secreted by them. This secreted galectin-4 binds to T cells inside of the tumor, causing apoptosis of these T cells. And we find when we look at tumors with or without galectin-4 expression that removing or if you have galectin-4 expression, you have a reduced survival and a reduced immune cell quantity inside of the tumor, as well as activity and cytotoxicity of the T cells that are still alive. In addition to this, we could also see that the binding site of galectin-4 on T cells is the CD3 receptor, and we even have a specific residue that we believe is the binding site of galectin-4. So when it comes to the immunohistochemistry of pancreatic cancer, we performed regular DAB stainings of normal tissue in both mouse and human, primary tumor, and the pancreatic cancer model system in mouse. We could see that there was a very strong stain of galectin-4 in the cancer cells. We also performed the DAB staining on metastasis, both in the liver and in the lymph node of human patients. And we could see here that there was also a very strong expression of galectin-4 in these metastases. And considering the function of galectin-4, it is probable that the galectin-4 expression is related for establishment to the establishment of metastasis by killing off immune cells trying to attack and kill the cancer cells. Interestingly, what we found when we performed the stainings on the human PDAC tumors is that we could find a light sheet of galectin-4 expression surrounding these so-called tumor glands. And we believe that this was secreted galectin-4 that was in very close proximity to these glands. We tried quantifying this, but we couldn't really get good reproducible data. So we started our collaboration with Lumito to use this UCMP method in order to try to find this out. So why did we choose to use UCMP? Well, formalin fixed paraffin embedded tissue is characterized by autofluorescence, but this is of course the most common tissue sample that you have available to do analysis on in a research lab. DAB staining is a tool that you can use, but this is saturated early, so this means that if you have twice the intensity of the DAB stain, it doesn't mean you have twice the abundance of protein. You can quantify this by scoring it by a human observer, but getting a semi-quantitative measurement of the intensity is a little bit more difficult. So we chose upconversion nanoparticles because they have low autofluorescence, they have a very low background in the stains, and then a high sensitivity for the targets that we're looking for. And you can also get a semi-quantitative measurement of the intensity of the signal, which was something that we were interested in. So in order to do the analysis of our tissue sections, we use QPath as a software. So this is an open source software that lets you do annotations and measure intensity in the pixels that are present in an image. We also perform staining on the normal duct of a normal pancreas to see what the general background was using this method, and we could see a very low signal in these normal stains as compared to the tumor stainings. So this is an exemplar image of a tumor that we stained for galectin-4, and we could see that there was a very, very strong signal in the epithelial cancer cells, and that it was surrounded by an area of lower intensity, which we believe to be the secreted galectin-4. So we proceeded to try to quantify this area of secretion. So this is how we performed the annotation on the tissue sections. So we generated an annotation surrounding separate tumor glands in the tissue, and we wanted them to be at least 100 micrometers removed from adjacent glands to avoid any spillover effects of secretion from both directions, so to say. We then generated these fixed area tiles that are 25 micrometers squared, where we could get a semi-quantitative measurement of the signal. We then annotated the three areas, epithelial, proximal, and distal, based on the distance from the epithelial cells. So this is how it looks as an example. So you have in green the epithelial cells, which has the highest intensity of galectin-4 signal. You have the red, which are proximal to the tumor gland, and you have the purple, which is the most distal ones in relation to this tumor gland. And we then could get a semi-quantitative figure for each of these tiles, and we could generate the signal for the abundance of galectin-4 in these three different areas: epithelial, proximal, and distal. So what we found was that the epithelial compartment, of course, had the highest signal of galectin-4, and there was a higher signal in the proximal part to the tumor gland as compared to the distal part of the tumor gland, which indicated to us that galectin-4 is secreted but only a very short distance in relation to these tumor glands. So interestingly, we also find that the secretion of galectin-4 varied with the differentiation of the tumor. So the initial analysis was performed on moderately differentiated PDAC, while we also performed staining on well-differentiated PDAC. But doing this, we found that even though you still had a very strong epithelial cell signal of galectin-4 in these tumor glands that were, it was a very powerful stain, we could not see any secretion here that was higher than the background as compared to in the moderately differentiated PDAC. So this formed a new hypothesis on how the secretion of galectin-4 actually occurs in these tumors, because that's not something that's currently known. So following this analysis of the tissue sections, we also performed a standard assay, an ELISA assay, where we looked at the abundance of galectin-4 in the serum of PDAC patients compared to normal controls. And we found that galectin-4 was actually detectable in the blood of these patients. But the caveat for this was that if you had a disease in the GI canal that was an inflammation, galectin-4 would also leak out into the blood of the controls. So those patients had to be excluded from the analysis. So to summarize what I've been talking about today, when it comes to the UCMP technology, it enabled high fidelity detection of galectin-4. And we were able to visualize and quantify the proximal secretion of galectin-4. And we also formed a new hypothesis of galectin-4 secretion. When it comes to the rest of the results in the paper, we found that galectin-4 induced apoptosis of tumor-infiltrating T cells by binding to the CD3. There was reduced activation and cytotoxicity of infiltrating T cells in the presence of galectin-4. And that reduced galectin-4 expression increases survival when we used a mouse model system of this disease. And with that, I would like to thank all the group members of Group Öland and our collaborating authors, which made this publication possible. And of course, our overseas collaborators as well. And this is my name and email address. Thank you.